All right, so we're going to do 3 Frayer cards today. The first one is going to be about DNA. DNA, the function of DNA, the job of DNA is it carries genetic information. It stores your information. So DNA is like a hard drive. The information is in it. We just need to read it. The information in our DNA is written in code. That code is written with the bases ATGC. So our code for our language is 26 letters of the alphabet. We put them in the right order, we get a message. DNA's code, ATGC, they put those letters, only 4, in a bunch of different orders, and they get messages. Messages that say, make curly hair, make green eyes, make earlobes not attached, right? DNA, make the enzyme to digest lactose. DNA has the directions to make everything in your body. The structure of DNA, the shape of it, it is a double helix or a twisted ladder, and it's made of monomers called nucleotides. We talked about nucleotides earlier, way back in Unit 2, but that is the monomer of DNA. We're going to talk in more detail about nucleotides, but just to have an image of it, a nucleotide is a sugar, sugar, a phosphate, and a nitrogen base. We have a sugar, a phosphate, and a nitrogen base. That's just the nucleotide. That's not DNA. When we take all these nucleotides and we put them together to form DNA, DNA is going to look like a double helix. I'll show you a pretty picture and then I'll draw it. Right? So the regular shape of DNA is a double helix. So when I draw it, because I know it's hard to draw from like a pretty image like that, I take one line, squiggle, 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 squiggle. The other line, squiggle, squiggle, squiggle. There we go. And my nitrogen bases go across the middle. We'll talk more about the structure as we go through class today of where the individual parts are found. We can take that DNA and we can organize it into either, I'll move this up here, either chromatin, which is loose, messy DNA. I'm going to draw that. Chromatin looks like this. Or we can organize it into chromosomes, which are tightly packed and organized. Again, I'll draw it. Chromosomes look like this, with a dot in the middle like that. We're going to learn first just about this DNA, but we're going to finish with the chromosome Frayer card for today just so we have it for future reference. That was a lot for DNA, right? We got its function, its job, we got its structure, and then we got a bunch of different images. We got the regular double helix of DNA and the 2 different ways it can look. It can look like chromatin, it can look like chromosomes, and that's going to matter when we talk about DNA in the cell cycle and what the DNA is doing throughout the life of a cell. If you need to pause it, pause it. I'm going to move on to the next slide. So the next one we're going to do is going to be chromosome, right, which is just a shape that DNA has. So the definition of chromosome, it is a structure made of tightly packed DNA and protein. And it's made of sister chromatids. So when I draw it, that's one chromatid. That's the other chromatid. That's the centromere that holds them together. Sometimes chromosomes are single chromatids, sometimes they're double. We'll learn about that when, again, we do more of the function. But what a chromosome is, it's just a specific structure of DNA. We only ever see a chromosome during the M phase. So if you think about the whole cell cycle, G1, S, G2, and M, the only time we see this structure is in the M phase. Otherwise, DNA is chromatin, chromatin, chromatin. So this chromosome is a specialized structure that only exists during the M phase. Non-example is chromatin. I'll move up here. Right, that's when the DNA is loose and messy. That's a prettier picture of a chromosome. That centralized part is the centromere, and each of these represents a sister chromatid. Sister chromatid. Sister chromatid. This is still DNA. DNA is still in there. The DNA that we know, that double helix, is all inside here. It's just super tightly packed up and organized. So the chromosome structure is just taking the DNA and squishing it into an organized shape.